Okay, we are once again assembled to talk about neurons and neurobiology. And today's topic gets down into the mud, as they say, because we're going to talk about how neurons communicate with each other, which, after all, is the whole point of the nervous system. If you want to get a signal from your head to your toe, you have to have a network of things acting in concert to do that. And those things are the neurons. And so today we're going to talk about how they perform that act of communication that lets you be you. And we're going to do this in 4 really easy parts. First, we're going to talk about what's going on in the neuron when nothing really is happening, which is called establishing the resting potential. And once you understand that, everything else is kind of variations on the theme. So we'll do the resting potential in some detail and then try to refer back to that and elaborate upon it as we go through the subsequent parts. So in part 2, we're going to talk about spiking an action potential, which is where the activity really takes place and changes. Then the propagation of that action potential once it's spiked. And lastly, this thing called synaptic transmission. Thrilling stuff, I assure you. Let's get started. So, let's take on, first of all, the big picture question of why the heck are we doing this? Why are we studying how neurons work? And maybe it's sort of obvious if you think about it for 30 seconds, but in the event that it isn't obvious, well, it's basically this. You have all these neurons in your system. You have a brain that has a couple billion neurons in it, and they can't be completely disconnected from one another, or otherwise they would have no overall function. And so the point is that these neurons do their wonderful things like helping you write poetry or read a book or whatever because they can talk to each other. When we talked about brain structure and function, we looked a little bit at the different parts of the brain and what's going on in those different parts. Well, those specific functions also require this communication. And the big things that we're interested in, hope and religion, definitely require a very tight level of communication between your neurons and other higher executive things like memory and futuristic planning and all that good stuff. So this can't be done unless the neurons can talk to one another. We'll also want to understand later on in the course how psychotropic or psychoactive drugs work, and that too is intimately connected to the individual neurons and how they themselves work. So we're going to spend this one last presentation on the nervous system talking about how this communication between neurons works. I mentioned you've got a gazillion neurons, and all of the important things you do every day, mostly without thinking about it, depend on those neurons having this functionality that includes the ability to communicate with one another. Now, you might think, oh my goodness, well, that could be a life study in and of itself, which in fact it could be, but most of you will not be neurobiologists. Sigh of relief, I understand. But I will let you off the hook in the sense that despite the fact that you have all these many neurons, all of them work basically the same way. There are some particular details that govern how specific kinds of neurons work, and this is realized primarily in the form of which neurotransmitter they use. So there is some difference there, and we'll talk about neurotransmitters later on so you'll understand how and why I brought it up. But the big picture is that the basic job of the neuron is simply to receive information from some other neuron and then process it and send it to another neuron in the chain. And that's what we're going to be looking at today. As I mentioned, it's easiest to start and get a tentative understanding of how this all works by looking at what's happening at a neuron when nothing is happening, when they are in fact at rest. And in this state, we say the neuron is at its resting state, and it is set up to receive and then to send electrical signals, but it is not doing so at this point. So we need to understand how the resting state is set up in the neuron, and then we will have a good grounding with which to start. So let's return to the basic neuron. You've seen the structure before, and it looks a little bit different in this graphic, but trust me, the parts are all the same. For instance, we have the soma or cell body. This is the thing you would normally associate with a cell. It's got the nucleus and the mitochondria and all that good stuff in there. We also talked about having branched processes that emanate from the soma. Because they're highly branched, we call them dendrites. And then we have this other thing, which is longer and skinnier and largely unbranched except at the terminal ends, and this is the axon. So we'll be talking about axons and dendrites quite a lot today. Now, another way to look at this whole process is that neurons work through the act of signal transduction. That is to say, they take a neurological message that starts out in one form, in this case an electrical form, and then in trying to get that message to the next neuron, the message is transduced into a chemical form, and then finally the communication loop is completed when the neuron sends the signal via chemical means to the awaiting next neuron in the chain, at which point the signal becomes, yet again, electrical in nature. So we're going to be talking about a message changing form as it moves along. But back to the resting neuron. In order to send and receive electrical signals, remember I mentioned that the signal starts out as an electrical signal and is changed to a chemical one before going back to an electrical one. Well, in order to send and receive electrical signals, currents have to be formed. And if this sounds horrible to you, like, oh gosh, I avoided physics like the plague, I'm with you on that. I did terrible in physics. But nonetheless, we can do this because it's going to be easy and it won't be that painful. All right, so in order to send and receive electrical signals, currents have to flow. And this is done in the neuron by setting up a charge differential across the axonal membrane. So we're talking now specifically about the axon part of the neuron and the membrane that surrounds that axon. And this in turn is done by using pumps and specialized gates that are present in the axonal membrane to set up this differential distribution of molecules that we call ions because they're charged. They have a negative or a positive charge. And so the axonal membrane, in conjunction with these pumps and gates, set up this differential distribution of charges, or ions, that will form the basis for conducting currents later on. So let's review what that membrane thing is all about. You know from your study of biology in high school, yes, you did have it, I know you did, especially if you were in Ohio, you knew that every cell is surrounded by a membrane, and that membrane had a really important function, which was to regulate the traffic of other stuff in and out of the cell. The cell wants to keep some things in, like those things it needs to live, and it wants to keep some things out, like those things that are toxic and might kill it. And so one of the jobs of the cell membrane is to make sure that only those things the cell wants to get in, and those things that it doesn't want to get in stay out. So we call that selective permeability, the ability to regulate traffic of different chemicals in and out of the cell. So let's review very basically the structure of a biological membrane, which you may recall is formed from two lipid layers, or a lipid bilayer, if you prefer. So here's one layer of this membrane. I'm drawing a red line through these round globules, which we call the hydrophilic heads. Hydrophilic because they love water. They're water soluble in nature. Now, there's another part to this half of the membrane, and that is formed by these squiggly little lines here. This is the so-called hydrophobic tail. Hydrophobic means afraid of water. So this is a lipid-based molecule, not a hydrophilic one that's water-soluble. And in order to minimize the interaction of these hydrophilic tails, which don't like water, with water, there's a lot of water outside, there's a lot of water inside the cell. So to protect them from having to interact with water, these hydrophobic tails are directed towards each other and are found on the inside of the membrane, which minimizes their interaction with water, which is thermodynamically unfavorable. Yeah, I did take physics, but I was terrible at it. Okay, so you have this bi-layer structure to the membrane. Here's the other layer. Here are the hydrophilic heads. Here are the hydrophobic tails. So that's the second half of the membrane. And projecting both out and through the membrane are these other things. You see a particular kind of protein called an alpha helix. You see a glycolipid over here. These are things that have specific functions like binding with antibodies and initiating immune responses, or allowing for particular things to move into the cell. The big red globules are generally proteins, and these proteins often serve as carriers, so they will allow specific chemicals to bind to this part of the protein, and then the protein molecule flips around and dumps whatever has bound to it inside the cell. And that's one way that things can get from outside to inside, but it requires a specific carrier, and only selective chemicals that fit this receptor can get in. So this is your basic bilipid membrane, and the reason I bring this up is that the axon of the nerve cell has such a membrane surrounding it, consisting of these two layers. Now, when neurons are at rest, they're going to have a specific distribution of those ions that I mentioned, those little molecules that have either a positive or negative charge, and they're going to have a different charge and a different set of chemicals inside the axon, inside the axonal membrane, versus the outside of the axonal membrane. So, if we first consider what's going on inside the axonal membrane, so we're inside, we will note the following. First of all, there's a whole bunch of this thing called K+. K stands for potassium. What? I hear you cry? Potassium doesn't start with a K? Well, that's true. But the word from which potassium came was kalium, which I think is Germanic, and it does start with a K. So that's why potassium, which begins with a P in English, has the letter K. But more importantly, it has a positive charge, so keep that in mind. We also see some negatively charged species inside the cell, particularly chloride. There's a whole bunch of it inside the cell at the resting state, and also a mother load of the thing we call anions. Anions are negatively charged species of various sorts. They could be individual atoms. They could be small molecules like amino acids. They could be a variety of other things that all have a negative charge. And so we have the designation A plus the negative charge. Okay? So that's what's going on inside the cell. To review, we have a mother load of this positively charged ion called potassium, and a mother load of negative charges primarily provided by these anions, but also by chloride ions as well. Outside the cell, we have a different situation. We have much, much less potassium, but on the other hand, we have a mother load of this thing called sodium, which is abbreviated Na. Why is that? I hear you cry. Well, back in the days when they discovered sodium and named it, they named it natrium. That's why we have an Na to stand for sodium, which starts with S. But more importantly, there's a positive charge associated with sodium. We do have some chloride ions outside, and because we're lacking the anions, it turns out that relative to the inside, there's a whole bunch more positive charges, largely coming from sodium, outside the cell, and there's an abundance, a hyperabundance of negative charges inside the axonal membrane, inside the cell. Okay, so we're going to do one more time in English. We'll review that again. To summarize, the resting nerve cell has much more potassium inside the cell, much more sodium outside the cell. That will be on a quiz, so make sure you know it. Next, there is relatively more negativity, more negative charges, largely coming from chloride and the ions, inside the cell relative to outside. So those are the two big things you need to know about the resting neuron. More potassium inside, more sodium outside, and overall the inside of the cell is more negative than the outside because of the anions and the chloride ions. And remember that this membrane surrounding the axon is selectively permeable. So these things cannot simply move across the membrane and escape either inside or to the outside. Big molecules like sodium and chloride simply do not move with ease across the axonal membrane. Plus, they're charged, which also makes it much more difficult to move. And so they're basically going to stay where they are until something else happens. The something else will be either the opening of a specific gate that allows passage from inside to outside, or the use of a particular kind of pump that exchanges sodium for potassium, but we'll get to that later. Okay, so because of this, because we have more negative charges inside relative to the outside, there's a charge differential of about 70 millivolts when you go from outside to inside. Now this charge differential is really important because remember I said we have to conduct some currents, and the currents require having different charges in residence. And if you don't understand that, do not worry, we will go over it again. Oh, infinite joy. Now, I did mention that there are ways for these charged species to move in and out of a membrane. They don't do it on their own. They don't do it without some help. And that help comes, first of all, in the form of gates, which can open under certain circumstances to allow these ions to move back and forth. So we have a membrane here, and you see that in that membrane we have this funny looking part here, which is one of those aforementioned gates. Now let's assume that these little black dots out here are sodium atoms that are positively charged, they're floating around outside the cell, and we now want to move one of these sodium ions inside the cell. Well, this sodium ion can bind to a receptor on this gate, which is the same shape as the sodium atom, and that's how the cell knows it's the right thing, and it's okay to open the gate and let it in. It has to have a specific size and shape in order to do that. And when that happens, when the sodium gate opens, these sodium atoms can move inside pretty much freely down a concentration gradient. Remember I said there's a lot more sodium out here than there is inside, and so if we're having free passage through this gate down a concentration gradient, it means that sodium will move from the outside where the concentration is high, inside where the concentration is lower. So sodium will move preferentially in that direction. And that's one mechanism by which sodium can enter a cell. So let's take a moment to pause and review. You should be able to tell me, or at least respond to a multiple choice question about the resting potential. What is it? How is it set up? Secondly, what is the distribution of ions during the resting potential? So basically, sodium outside, potassium inside. We want you to be able to describe the distribution of charges during the resting potential. So you would say something like more positive outside, more negative inside. And we want you to be able to understand how that resting potential is maintained. And of course you know that because it's that axonal membrane which is selectively permeable and only lets specific things in at particular times. In this case, we talked about the sodium gate that does that. Okay, moving on. Okay, so now we're going to talk about the next step in the transmission of a nerve signal. And this is given the name spiking and action potential. So something new and something different is going to happen. We're going to move out of the zone of the resting potential and into this active function, which is spiking the action potential. And this is done because the nerve cell needs to be able to send and receive electrical signals along its axon. And this is the mechanism by which it does that. So we're going to start with the question, what happens when the resting neuron receives an electrical message from another neuron? And the reality is that the dendrite is the first place at which that electrical message is received. And that the dendrite has the ability to, through an electrical means, send it to the axon. And we're not going to detail that in this particular lecture because you don't really need to know that. So we'll gloss over that and get to the real activity of importance to us. And that is that once the axon has been communicated to by the dendrite and knows that there's an action potential, what happens is the axonal membrane becomes less resistant to the passage of sodium through the membrane. And this is done by opening up those sodium gates that I mentioned earlier, and then the sodium moves into the cell along a concentration gradient. And this has a really important effect on that resting neuron. Namely, it reverses the distribution of charge across that membrane, but, and this is really important, only in a localized area. So in the resting membrane, there's more positive charges outside, more negative charges inside. But when an action potential is going to be spiked because of the influx of sodium, there's a charge reversal in a small area. And in this small area, the charges have been reversed. So there are more negative charges outside, more positive charges inside. And we call that condition of having those charges reversed depolarization or depolarized, but only in that small localized area. Again, if you don't get this, fear not. We'll go over it again. Now, some of you are accountants. I've looked at your degrees, or what you're getting a degree in, and some of you are, in fact, accountants. So for those of you who like to keep track of pluses and minuses, I will acknowledge to you at this point that after receiving an electrical signal and having the influx of sodium, following that, there's a time during which the axonal membrane becomes permeable to potassium, and the potassium gates open, and potassium moves outside the cell down a concentration gradient. But this takes a little bit longer, milliseconds longer, to happen than it does to have the sodium gates open. So for a time, a short time, and in a limited part of the membrane, there is this charge reversal that I mentioned, and we gave the name depolarization. All right, easy peasy. And this now is set up for an action potential to be spiked. Action potentials are simply the electrical messages created and conducted by the axons. And once the depolarization that takes place because of the influx of sodium into the axon, once the depolarization reaches a key threshold level, then an action potential is triggered. And this then allows for current flow with the next area of the membrane, which is still at the resting state and therefore is oppositely charged. And when the current flow reaches that next area of the membrane, the same thing happens, namely the resistance of the membrane to sodium in that next area, which is adjacent to the depolarized area, begins to recede. So the membrane opens, the sodium gate opens, and sodium begins to flux into the axon at that second site, and we get a charge reversal in that second site. And using this mechanism, the action potential moves down the length of the entire axon. Remember, axons are quite long, and so if the the action potential starts near the cell body, the action potential has to get down to the end of the axon in order for the communication with the next neuron to take place. And this is the mechanism by which the action potential will sweep down the length of that entire axon. So here's the graphic that kind of lays that out for you. So the action potential, which is abbreviated for you AP in this particular graphic, the action potential is the site of the initial depolarization. So this is the area where the receipt of the message from the dendrite causes the sodium gates to open. The sodium atoms begin to move in. This makes the outside now more negative relative to the inside. And as a result, we would consider this section of the membrane to be depolarized. Now, if you look at the distribution of ions here, where the action potential is spiked, and the subsequent section of the membrane, which is highlighted for you in blue, you will see that it is still at the resting potential because there are abundance of positive charges outside and negative charges inside. Because we have opposite charges across the membrane in these two locations, we can now have current flow, as indicated here by these arrows, between the pink area and the blue area. It's the charge differential, which is different in these two places, which facilitates the current flow. And the receipt of the current in this second area of the membrane, which is currently at the resting potential, has an important effect. Namely, the receipt of the current here causes the resistance of the membrane in the blue part to become less to sodium. And so the sodium gates open up and sodium fluxes in. We get a reversal of charge here in the second area of the membrane. And so now we can have current flow between this second area, which is currently in blue, with the next area of the membrane. We'll say it's over here. So in this will be the new localized area. And the receipt of current over here results in depolarization in this third section. So this is our third section of the membrane. This causes the depolarization, which then allows current flow between that and the next area of the membrane. And so it goes all the way down the length of the axon till we get to the bitter end. So I've just described part 3 for you. That was thoughtful. Namely, how the action potential is communicated down the length of the axon so that the message can be sent to the next neuron. So to recapitulate, we have current flow taking place between the depolarized area of the membrane, which is initially was the pink area where the action potential was spiked. And then we get current flow between that pink area and the next section of the membrane, which was highlighted in blue. It was still at the resting potential. The receipt of the current in that second area, the blue area, causes sodium to rush into the second part, which was in blue. That reverses the distribution of charge across that blue part of the membrane. And now we can get current flow taking place between what was the blue part and the next part of the membrane, resulting in depolarization and then current flow. And then it just continues, depolarization and then current flow down the rest of the axon. So here is a graphic that shows you electronically what's going on. We mentioned initially that in the resting neuron, the charge differential between inside and out was minus 70 millivolts. And then, once the news of the receipt of the action potential by the dendrite is delivered to the axon, the sodium gates open. Like I said, sodium begins to move in, and as a result, the charge differential begins to move towards zero. So at this point, there is no difference between the inside and the outside as a result of the sodium having moved in. And here's another interesting thing. The movement of sodium inside the axonal membrane is one of the rare examples of positive feedback in nature. Normally, natural cycles work on a negative feedback basis. But this is one case where we have positive feedback. So as the sodium moves in, it causes for the amount of sodium fluxing in to increase as time goes on. And as a result, not only do we get to zero, there's no charge difference, but as the positive feedback plays out and more sodium moves in, the potential overshoots zero and enters a positive zone. In other words, at this point, the inside is 30 millivolts more positive than the outside. Now, eventually, these sodium gates will close, and potassium gates will open. So potassium will flux out of the cell at this point, and electronically, that seems to fix our problem. It goes back to minus 70 millivolts. But eventually, we're going to have to restore the resting potential, and that means shuffling these ions around. The charge differential has gotten to the correct level, but we So let's recapitulate what you need to know. You need to know a little bit about how the action potential is created, namely that it's received by the dendrite, communicated to the axon. You need to know what happens to the distribution of ions across the axonal membrane after the action potential is triggered. You need to know that there's current flow between adjacent areas of the membrane, and that current flow triggers a series of events in which sodium moves in to the localized area, reverses the charge, sets up current flow, and the receipt of that current by the next section of the membrane also then results in depolarization, etc. All right, let's assume now that our action potential has gotten to the end of the axon. Now we need to talk about how the signal gets transduced into a chemical form and how that chemical form moves across the space between axons and initiates a new action potential in the next neuron. So we need to talk about how the signal is transferred from the first neuron to the next neuron in the change, as well as how an electrical signal gets converted to a chemical one and then back to an electrical one. The chemical signal is provided by a substance known as a neurotransmitter. The name itself should pretty much tell you what it's doing. It's transferring a neurological signal to the next neuron. So to understand this, we need to look at what's going on in the place where two neurons meet. So here's neuron number 1, here's neuron number 2, and what we're saying is that this neuron has received an electrical signal, it has spiked an action potential, it has sent that message down to the end of its axon, and the electrical message now is here waiting to be transferred to this next neuron. And in order to make more sense of that, it's helpful to blow this up and look at it in a slightly larger form. So this is the terminal end of the first axon, the one that has received and sent the action potential to its terminal end. This is the next neuron in this sequence to which the neurological message is going to be passed. And the mechanism by which this happens is that the receipt of the action potential down here at the end of the axon of the first neuron causes one of these little round vesicles, which is filled with a specific chemical called a neurotransmitter. It causes the membrane of this vesicle to fuse with the axonal membrane. So here you see that product. The vesicle containing this neurotransmitter has a membrane around it, and its membrane has fused with the axonal membrane, which means that it dumps the neurotransmitter molecules contained within that vesicle out into this space between the two neurons, which we call a synapse. The space between two neurons is called a synapse, or we can also refer to it as the synaptic cleft. It simply means the space between two neurons. So now the business of converting to a chemical message has taken place. The electrical signal causes a specific amount of this neurotransmitter stuff to be released into the synaptic cleft. The neurotransmitter substance diffuses across this space, across this cleft between the two neurons, and then the neurotransmitter molecules will bind to specific dendritic protein receptors on the dendrite of the next neuron. And the receipt of this chemical messenger will then initiate a new neurological message in this second neuron, which will be sent to the axon of this neuron, where an action potential will be spiked. And of course, the message that we're talking about, the one that's being communicated from one neuron to the next, is chemical when it enters the neurotransmitter phase, but after it really binds to the dendritic protein receptor on the second neuron, it again becomes an electrical signal. All right, so this is really important because when we study things like depression and how drugs that relieve depression work, we'll be coming back to the neurotransmitter story again and again. Not only that, but the neurotransmitters can also serve as neurohormones, and they have a huge role to play in hope and belief. So, the phenomenon of releasing neurotransmitter substances that diffuse across the synaptic cleft to then initiate a new neurological message in the next neuron is called synaptic transmission. And this takes place, again, so this is a review, after the action potential in the first neuron reaches the end of neuron 1. It causes those vesicles filled with neurotransmitters to fuse with the axonal membrane, causing the neurotransmitter substances held within the vesicles to get dumped into the synaptic cleft, or the space between the two neurons. The neurotransmitters then move across the synaptic cleft, or the gap, and bind to dendritic protein receptors on the second neuron, which then initiates a new series of electrical activity culminating in an action potential in the second neuron. And that's how it happens with all of the neurons in the change until the ultimate destination, whether it's a muscle or a gland or something else, receives a message and responds appropriately. So, sounds like we should be done, right? Well, we almost are. But remember, we left our depolarized axon in a depolarized state. And in order to receive and send additional messages in the future, the first neuron has to do some cleanup activity, which involves repolarizing its membrane so that it can restore the resting potential. So basically what it needs to do is swap out all the sodium that moved inside the axon for some equivalent potassium that it moved outside the axon. And this takes place during a finite, although brief, period of time called the refractory period, during which the cell cannot conduct a new action potential because its charges are all messed up. So there's a special enzyme that lives in the bilipid membrane that we talked about earlier, whose job is to exchange sodium, located now in abundance on the inside of the cell, for potassium, which is now currently in abundance on the outside of the cell. So it does this, and that's the way it restores the resting potential so that the neuron can receive and send additional neurological messages. So, to get through productively this part of the discussion, you need to know what a neurotransmitter is. You need to know how and where it's released. Again, we're going to be talking about this in subsequent lectures, so you really, really do want to know this. And you need to know what happens to the neurotransmitter once it's released. We'll talk about this in more detail when we get to the discussion on SSRIs. So we'll have a specific example to help you recall all of these things. And here, blessedly, is your study guide for this final series of discussions on neurons and what they do. So the rest is easy. You can do this. All right. See you next time.